think this is a very important talk, so please pay attention. I think Dr. Arka Ghosh has done some extraordinary work in building up the GROW laboratory at Narayana Nethralaya, and they're on the verge of some very important translational work. So Arka, it's all yours.
Thank you for the introduction, Vijay, and thanks for the invitation to talk here. So I will speak less about the science we do and focus more on some of the translational aspects of the work we do that can hopefully benefit the patient community in India. So this is a slide just to tell you about what genetics has done for rare disease and diseases in general. So each circle here that you see lists the number of genes that we know for a given disease, right? So for all the genetic disorders, almost we know at least a few of the genes that cause that disease. So once we know the genes, we should be able to treat it, but it's not that simple. And that's the part that I'm going to address and I'm going to talk about. So essentially, what is gene therapy? If you know the gene that is mutated in the disease, you essentially take the normal copy of the gene and package it into a delivery system. So here I'm giving you an example of adeno-associated virus because that's what I've worked on. And you package that gene into this virus, and this virus is used as a delivery vehicle. So it goes to the site of the disease and expresses the right protein, and therefore you reverse the disease. So it's fairly simple. Only in theory, it's very, quite troublesome to execute. Which is why, although this was theorized almost 40 years ago, with once the structure of DNA was known, and once we actually knew which genes are causing which diseases, but it took this long for us to get to actual therapies. So the therapies that are normally done, and it really depends on the kind of disease that you have. So the most common one is to put a normal copy of the gene to compensate for the non-functional copy of the gene that the patient has. You can trade an abnormal gene, so this is more on the recombination front. An abnormal gene can be repaired through a reverse mutation or alternate splicing, which is what CRISPR is trying to do. It will go in, fix the mutation, make it normal. And the other thing is change the regulation of genes. So this is the kind of gene therapy that is most likely going to be the first ones to hit the market, simply because if you look at independent genes that are mutated in any given disease, the number of patients is very small. So it doesn't make economic sense for companies to put in money for that. But if all those genes are regulated by one overarching mechanism, and if you can kickstart or modulate that mechanism, then that becomes very, very attractive because your pool of treatable patients becomes much larger. So I'm just going to show you some data from genetherapy.net, or if you can go and check. So these are the kinds of vectors that are in clinical trials, the distribution of it. And the gold standard, I'll tell you why, is obviously the adeno-associated virus. Geographical distribution, so as you can see, it's by and large in the United States. And this is where the sad part is for India. Of the 70 odd countries that participate in gene therapy trials, India does not figure in that yet. So we have to quickly change that. Again, the different kinds of indications. Bulk of the gene therapy efforts have been in cancers, and a lot of the available therapies are also for cancers, and I'll show you a list of those. And of course, by phases, if you look at bulk of the therapies are still in phase 1. Again, something that has been pointed out by speakers before me, for rare diseases, and this Kiran also mentioned, for rare diseases, if you don't have that many patients, you can't have phase 1, phase 2, phase 3, phase 4, you have to cut down on those things. So So here's a list of approved cell and gene therapies. This is not an exhaustive list, but sort of a curated list from FDA and European websites. All of these, of course, are for hematologic disorders, which has always been the most advanced in adopting these things. I'm not going to tell much about this, but on this side, you have quite a few of these therapies that have been approved in the last year and starting from 2015. Of course, Luxturna is something, so we work primarily in the eye field. It's for retinitis pigmentosa. Glybera is for LPLD. So this came out in 2015. In Europe, it got approved. But this also costs $1 million for a treatment. So it is now at risk of getting pulled back because there aren't that many takers for it. Gemdizin is the one that's been the flag bearer of it. This is for head and neck cancer. It's very simple. It's an adenovirus delivering P53. It's been there for more than 8-9 years now. And of course, Yescarta, this Sharad talked about. And what you will notice is that most of the approved ones, there's a lot in cancers, right? So for monogenic disorders, things have always been slower. So to prepare for a trial, you need to select the kind of vector. So like I said, we know the gene, but how do we take the gene and send it to the right place, to the right tissue, to the right cell types? So a variety of different kinds of vectors are available. You can have viruses, which I'll talk more about. You can have non-viral delivery systems, which are cationic polymers, dendrimers, cell-penetrating peptides, liposomes, and now there's naked DNA also. So these are sort of the driving characteristics of a vector delivery system that we need to consider when we are trying to decide which patient will be suitable for what kind of therapy. So essentially, it should not cause toxic reactions. It should be present for a long time. You want to take a therapy that lasts you for at least a few years, not having to take an injection every day or every few months. So that's where viral vectors are at a huge advantage. So there are a variety of viral vectors that are used for gene therapy in trials as well as particularly in the labs as of now. But adeno-associated virus has proved to be the gold standard so far for a number of reasons, and I'll allude to that here. So first, it very efficiently delivers your gene to both dividing and non-dividing cells. This is very, very important because if you look at neuromuscular disorders, your muscle cells, your neuronal cells, they are typically, they don't keep dividing, okay? They have reached the end of their division cycle. So lentiviruses, for example, or gamma retroviruses, they often need the cells to be dividing for them to be incorporated. Second, it does lead to pretty high levels of gene expression. And there is a complete absence of viral genes in the recombinant genome. So this is a cartoon of the recombinant genome. So you have your transgene, the gene that you want to put in, and nothing else. There is no viral gene there. So you do not have a chance of the viral proteins causing an immune reaction. One important thing about AV is it is not associated with any known human disease. So right off the bat, it's very, very safe. The prototype AAV virus, although we do carry a humoral response to it because we are exposed to it in childhood. So absence of significant toxicity and immune reaction is one of the primary safety profiles in this vector. So now I'll show you some of my own data. So this is at the time when I was working on muscular dystrophy. So a single infusion of the viral vector can cause body-wide gene expression. So this is a sham injection, and this is an alkaline phosphatase gene. It's a reporter gene. So wherever you see dark color, you know that the gene has expressed. So what you can see very clearly is that almost every fiber in the body, in the diaphragm, in the heart, all muscles, they get they accept the virus and they express the gene. So it is theoretically possible to treat even devastating diseases like Duchenne muscular dystrophy, where every muscle type in your body is affected. We also tested these vectors for immune reaction, and this is in the MDX mice, which is a model for muscular dystrophy, and the virus, the vector does not cause any additional immune problems. So remember, the reason we are checking it in a disease model is in a disease model you already have a lot of hyperimmune reactions happening, a lot of necrosis and fibrosis, all those kinds of things happening. So it does lead to a reduction in gene expression. So in normal wild type cells or wild type animals, you see a much higher level, but the level that you see in diseased animals is still clinically significant. Then we moved on to larger models. So these are beagle dogs, and we also did it in a naturally occurring mutation in a golden retriever dog. So again, all these skeletal muscles are very well transduced, but there is a window in which you have to do the therapy, and this is very important. This even Anu is going to talk about that why a therapeutic window is so important, which is why it's important for us researchers to get these things online as soon as we can, because the disease can only be checked in a certain window. So this is an evidence of that. Adult dogs treated with the therapy and neonatal dogs, and you can see all the fibers are expressing the gene here, but not so much in the adults, because the immune reaction is clearing things off. And that's going to happen. So I work in the eye, so obviously I'm going to show you a little bit about how well these vectors work in the eye. So this is alkaline phosphatase, this is a sham injected eye, and you can clearly see the retinal layers. Here you can see the plexiform layer very nicely. So the photoreceptors get the gene that you are providing. This is more recent work. This is for corneal haze, so blindness, preventable blindness if you would, and this green here shows the haze, so that prevents you from seeing, so that gets considerably reduced when you do the gene therapy. So we do work on retinoblastoma. This is a rare cancer, and again there's a need for therapies in retinoblastoma because we have much lesser cases. It's 1 in 15,000 to 18,000, so there aren't any specific targeted therapies. In fact, in retinoblastoma, there's not enough agreement between the clinicians as to what should be the chemotherapeutic or radiotherapeutic treatments given. So, but given our population, we have over 1,500 cases a year. A lot of these cases end in patients not surviving. This is how the tumor looks inside the eye. So by the time this tumor grows and comes out of the eye, the patient will not be surviving. So it's important to understand. So is gene therapy a possibility for retinoblastoma? So this is just a snapshot of the studies we did. So a normal retinoblastoma cell can form these tumors, but if you put back the retinoblastoma gene, it can no longer form the tumor. And you can see that in the tumor counting. So it is possible to do gene therapy even for pediatric cancers. So that's the point I wanted to bring across. So now a few discussion points I want to bring forth is what's important, what is currently being developed at a rapid level, but it needs to go outside of big cities like Bangalore or Mumbai, is to identify clinical teams that can have the infrastructure for running gene therapy trials, because these are very long trials. The patient's follow-ups have to be in years, and you have to have trained personnel for those kinds of things, and they should be ready for surprises, because a lot of these we don't know what our in our genotypes what the response to a therapeutic vector would be. So our immediate hurdles, apart from the regulatory guidelines, which I'm sure Vijay will talk a lot more about, is the vector production, making the actual gene therapy drug. So we need to have a CGMP production facility which requires class 100 clean rooms. Now these clean rooms are different from what are present in pharma, Because in pharma, those are all fully automated. There's no human personnel entering it. There's a bunch of machines that are doing that. In case of a gene therapy vector, it's actually going to be humans who are going to be making it. So it's that much more difficult to maintain class 100 specifications. From the patient point of view, we have to prepare for a gene therapy trial, which means we have to screen patients for what kind of vectors can be used in which kind of patients. That requires neutralizing antibody testing, which is not done, has never been done in an Indian scenario. So that is something that's very, very important, particularly for neuromuscular and blood disorders. So we've been building a gene therapy vector core. So this picture is a few months old. It looks much better than this now. And around this we've built a team of people. So we have our advisors, Professor Shomi Bhattacharya and Professor Kumar Manikaral. So Professor Shomi is the one who started the first, he discovered the first retinal genes in retinal dystrophies and went on to start the first gene therapy trials in mood fields in the UK. So he advises us. Our team of scientists and postdocs and students who are going to make the actual vector. Anu is here today. She's our genetic counselor and maintains the registry of patients. Of course, all our other support staff. So to summarize it, a variety of vectors are now available. I showed you a list of approved gene therapy products, and there's at least 2,000 different trials happening. Sadly, not a single one in India. We have to remediate that for sure. AAV definitely is something that, from a gene therapy point of view, rather than putting money in many different kinds of vectors, we should go with that. That's the gold standard. Across all the approved monogenic therapies, it's AAV which has been used. So with that, I would like to thank all my funding, my team members, and our lab. Thank you.
So when will the first gene therapy trial happen?
Hopefully by end of next year we'll be ready to run a trial. But of course, the government is allow us to do the trial.
That gives us enough time to get these guidelines.
Yes.
Okay, good. So thank you very much.